नमस्ते, हमेशा खुश रहो बिटिया. This is Dr. Lily Trivedi, and today I am going to give a lecture on my cell division, that is mitosis and meiosis. Now, for cell division, we have the cell cycle. Now, cell cycle is the series of events that take place in a cell leading to the division, that is replication. In cells of prokaryotic cell, it occurs by binary fission, whereas in eukaryotes it is divided in between two periods, that is interphase, where the cell grows and it accumulates nutrients for mitosis and then duplicates its DNA. There is the mitosis phase in which the cell splits into two distinct cells. Now they are called as daughter cells. And finally, there is a phase cytokinesis where the new cell is completely divided. Now, the cell division cycle is an important process in which a single cell egg or cell can develop into mature organism. So we have this. Now let's come to the next part of cell cycle. As over here you can see the cell cycle, there is an interphase and mitosis. Now this, here we cannot see the chromosome because they are uncoiled. Now as soon as coiling will take place, where it will take place, here the first phase which we take is the interphase. Now this is the majority of the time where the cell spends it, and the functions it perform is preparation of cell division. Now there are two stages. First is G1 stage, S stage and G2 stage. Now in the G1 stage the cell will grow and function normally, and protein synthesis will occur and the cell will become double in size. So if the cell is not dividing again, it will remain in this phase. After the G1 phase, there is synthesis phase in which the cell duplicates its DNA. It is also known as Swanson phase. What happens over here? There is semi-conservative mode of replication. You must have studied semi-conservative, that is in a zip-like fashion the DNA splits and it doubles itself. In the G2 stage, the cell resumes the growth and starts preparing for mitosis. Now what is mitosis? It is a process by which a eukaryotic cell separates the chromosome in a nucleus and two identical sets are then formed. So two nuclei are there and the cell divides, forming two equal daughter cells. So each daughter cell is similar to the parent. The first stage of mitosis is prophase. So here what happens? The chromosome starts coiling, the nuclear membrane disintegrates and the spindle fibers start appearing. After prophase we have the metaphase, in which the nuclear membrane is now completely gone. The chromosome becomes aligned in a plane. At anaphase, the chromatids separate and the number of chromosomes double. They are pushed and pulled toward the opposite poles by these spindle fibers. The last stage of mitosis is telophase, in which the cell divides into two. The chromosomes start uncoiling, the nucleus is formed again, and the spindle apparatus disassembles. So here this is the last stage of mitosis. Another type of cell division is meiosis. It is a special type of cell division necessary for sexual reproduction in eukaryotes. The cells are produced by meiosis as gametes or spores, and in many organisms including land and plants they are called as sperm or egg cells. First is reduction division, also known as meiosis 1. So meiosis 1 is known as reduction division. So a regular, suppose 46 chromosome is there in 2n, so each n will contain 23 pairs. So let's start with the first phase, that is anaphase 1. Then we have, there are two, meiosis 1 and meiosis 2. Now let's start with the first meiosis 1. The first phase is prophase 1. Now there are 5 substages: leptotene, zygotene, pachytene, diploneme and dikinesis. Now during leptonema, what happens? Chromosome condense and become visible. Then in zygonema they become dense and start pairing, and this is they start forming the synaptonemal complex. It is the complex which is formed between the two pairs of chromosomes. Coiling and shortening continues as the chromosomes become condensed and the synapses formed between the pairs, and it forms a way of tetrad. The sister chromatids in the diplonema stage becomes to separate slightly, now revealing the points of chiasmata. What is chiasmata? It is the point where they are joined, and this is where the genetic exchange will occur between the non-sister chromatids, and the process is known as crossing over. After the crossing over comes the stage dikinesis, where the chromosomes continue to pull apart, but non-sister chromatids are still associated through the chiasmata, and it begins to move towards the end of tetrad as the tetrad separation continues. So that is the last stage of meiosis, that is prophase 1, terminalization. And during dikinesis, the nuclear envelopes break down and spindle fibers begin to interact with the tetrad. So here you can see, this is a diagrammatic representation. This is they are becoming visible. They begin to start pairing, and the whole, they form a symptonemal complex, and these stages are known as the chiasmata, and again they are doubling. So after prophase 1 we have the metaphase 1. Now what happens here? The centrioles are present at the opposite poles of the cell. So these homologous chromosomes, that is bivalent, are tightly coiled and condensed as they will be in meiosis, and they become arranged on the plate called the metaphase plate, and the spindle fibers will contract, and here we are seeing the fibers are from the opposite pole till the homologous chromosome. After this we have the anaphase 1. The two chromosomes again start, they separate from the tetrad and become bivalent. That means two arms are there. Tetravalent means four arms are there. And they start moving towards the opposite poles of the cell as a result of the contraction of the spindle fibers. After telophase 1 we have the prophase. Now here we have, okay, we were in the anaphase 1. Then we have the telophase 1. Now what happens here? Complete migration of homologous chromosome has taken place, and haploid set of chromosome here is present at each pole. Then a nuclear envelope will be formed around each chromosome set, and the spindle fiber disappears and cytokinesis forms. Now after this we have the prophase 2. Now prophase 2 is duplication of which has taken prior to meiophase 1. Now here we see two cells are formed. Now again the centrioles will duplicate and they will separate, and two pairs of will separate into two centrosome, and again nuclear envelope will break down and the spindle apparatus will start forming. So after meiosis 1, the meiosis 2 is similar to mitosis. Then after prophase we have the metaphase 2. Now you can see both the meiotic spindle is there, it has separated, the metaphase plate has been formed, and the chromosome have arranged themselves along this plate. Now each is attached to the microtubule that is coming from that pole. Anaphase 2, the centromere will start separating and the two chromatids of each chromosome will move towards the opposite pole, and now they are called chromosomes in their own. After separation, they are now separate chromosomes. And when here again they are forming telophase 2. So we have four daughter haploid cells are formed. First was meiosis, that is reduction division, and second was mitosis, that is normal division. So nuclear envelope is again formed around each set of chromosomes. Cytokinesis is taking place. Each set has haploid set of chromosomes. Crossing over has taken place and there is recombinant cells segments in the original parental chromosome. So students, this was the brief about mitosis and meiosis. I hope you have liked my lecture. If you have any query, please give in the comment section. I will resolve it soon. This session was powered by Digital Version 2.0 of Jyoti Vidyapeeth Women's University. Namaste.